All right, so with mineral nutrition, we have the NPK is what you buy in the store. If you want fertilizer, it's usually NPK is sold as a mixture. Most plants, they actually prefer nitrate, NO3 minus. That's how they take up their nitrogen from the soil naturally. Some as ammonia or ammonium. NH4 plus is ammonium. NH3 is ammonia. If they take it up, it gets reduced usually to ammonium quite quickly, and then that usually gets put into amino acid production. Yes.
Why wouldn't it be the other way
around? I think it has a lot to do with soil systems and how plants evolved naturally to take up minerals pre-ag days. You know, nitrate was available. Of course, it's negative, so you know it's not stuck to the soil particles, right? So that's free in the water solution of the soil. But I mean, there are lots of plants that will take up ammonium. When I say most prefer, I'm typically, when you read that kind of resource, it's plants that they're thinking about that humans use. So a lot of it is crop production. Okay. On the other hand, I think like cranberry plants would prefer ammonium, so it varies. So if you're looking for nitrogen deficiencies, you are looking definitely for stunted growth. You will see an obvious difference in the biomass of the shoot and leaves and chlorosis. It's usually general chlorosis. If I just say chlorosis, that means general chlorosis. Once it gets into the plant, it is mobile. So if it's mobile in the plant, if a young leaf is saying, hey, I need more nitrogen, send some to me, what part of the plant's going to display the symptoms first? Older leaves. Older leaves will be going, oh, okay, I'll send you some. And then the older leaves, if they don't get that replaced and there's deficiency, they'll start to show the chlorosis first. So look on the plant body, old leaves versus new leaves, what's going on. I can tell you a lot. Another thing, not all plants do this, but tomato is a good one. Beans we're going to work with beans. They don't really do it very dramatically at all. Tomatoes do though. If you grow tomatoes in your garden and you notice a purpley look to the stems, or if you turn the leaf over gently and look at the underside of it's purpley, has a purple pigment, that's actually it's not just for nitrogen. There's a couple of different deficiencies, but nitrogen could be the culprit, a deficiency in that. And so it's an anthocyanin pigment that's accumulating. And so look for that. So nitrogen, super important for the plant, of course, because they need it for all the amino acid production, protein. So here you're seeing the strategies for some form of nitrogen coming into the plant. We have the options of ammonium, ammonia, nitrate. Okay, and you can see in at least some of the cases, it might be necessary to expend some energy to get enough nitrogen in. And so sometimes the plant will have to do that, use up some of their precious ATP. Okay, but they can take up different forms. It just depends what's most readily available. But nitrate, NO3 minus, is the dominant favorite form.
Do they reduce them?
Yeah, they do a quick reduction all the way down to usually ammonia or ammonium.
Oh.
But nitrite is one of the intermediate products. It just keeps going.
After nitrate or before?
They'll take it in as nitrate, okay, and then then they'll reduce it to nitrite, and they'll keep going. Okay. Yep. So phosphorus is another one on the NPK bag of fertilizer. And if you have typical acidic soils, normal, pH 5.5, 6.5, it's the H2PO4 minus form that's easily taken up. If you go more alkaline, then it is shifted, phosphorus is shifted into the HPO43 minus form, which is much more challenging for a plant to get access to in the soil. And so virtually unavailable is a good way to phrase that. So again, maintaining 5.5 to 6.5 is ideal for phosphorus, but you go above and it can become unavailable. And remember, if you go too far below, it can complex with, for example, aluminum and become a precipitate. So going below isn't good either. You want to stick with 5.5 to 6.5 pH where phosphorus is most available. You go below or above and it quickly becomes precipitated out, complexed, unavailable. So the amount of soluble phosphorus, when I say that, I mean that's the way the plant can get access to it. If it's soluble, the plant has access to it. That means it's soluble in the water. If it's precipitated out, the plant can't easily get access to that. So if it's bound also up in organic forms, there is phosphorus in decaying material, decaying organic matter, but again, they can't readily access that. And so if you ask a soil chemistry expert that works with plants and all that, they would say phosphorus is probably the most challenging for a plant. Of all the macronutrients, phosphorus is the most challenging for them to get access to because it's so easy for phosphorus to become unavailable in many different situations in the soil. So here, just if you go below the pH, I just wanted to remind you that it can complex with aluminum or iron and become, if it forms a complex with them, it's insoluble. It won't work for the plant. Basic soil, again, if you go way above pH 7, it becomes precipitated out. So it's really challenging. And so the symptoms, it's kind of weird. You'll look at the leaf and it looks intensely green. Instead of going chlorotic, it looks intensely green, at least initially. And then if the phosphorus deficiency intensifies, it'll shift into dead tissue. And this one can also do anthocyanin on the stems. That purpley look to the stems or the underside of leaves can also be due to phosphorus deficiency. And once it is in the plant, it is mobile. It can get sent to younger parts that are demanding more. So that means the older plant parts, the older leaves will display symptoms first. So phosphorus, there are some plants, because this is such an issue, phosphorus is needed in high quantities by the plant, and yet so often it is precipitated out. So there are a few strategies that some plants have adapted to, but not all plants. And so these diagrams are trying to show some options. So here on the top left, you're seeing that the plant, just through its normal respiration, just like what we're doing right now, we're exhaling out CO2. Plants do that as well, so to speak. They release CO2. If the roots do that, it can form a bicarbonate ion, which can help solubilize phosphorus that's precipitated or in organic form. So that's more of a passive mechanism, and often what has to happen, in addition to giving off CO2 just through respiration processes, they'll have to expend some ATP. So they're trying to show that ATP is being used to create a proton gradient because you need that proton to drag in, so to speak. It's a symport process. You need that proton energy, that energy of it diffusing to get the phosphate to be brought in. So it's costly to the plant to do it that way, but at least they can do it. The other one on the top right is looking at citrate. So they'll make citrate and then they'll secrete it. And again, that can help solubilize the phosphorus. And then the lowest picture here is an enzyme that they can make, excrete it out. And again, it can help solubilize. So there are some plants that can do this, but it's not, again, in the agricultural realm, very few plants in the agricultural realm can do this. And so the way they can get access to all this phosphorus that's precipitated or complexed is through the help of microbes. If the microbes are in the soil, they can do it. They can get access to the precipitated out forms of pretty much any of these nutrients. And as the microbes are breaking that down, releasing it from the soil, releasing it from the organic matter, then it can get released into the water, dissolve, and then the roots can take it up. So again, another reason why the microbes are so important. So I put this here to emphasize that point. There's a company called Jord Bioscience that my postdoc advisor actually helped form. And so my postdoc advisor, Linda Kinkle at the University of Minnesota, she has, and when I was a postdoc there, we were working on using beneficial microbes to help plants grow better, whether it's to fight off pathogens or to get better access to nutrients. And so she reached a point back in, I don't know if it was 2013 or 14, where she was getting frustrated with, we were finding all these great microbes, but it wasn't getting translated into the real world application. And so she decided to start a business and get these microbes out where people can start using them and getting better crops, less, plant diseases, more nutritional crops. And so I just think it's a fantastic story of, taking the academic research, so to speak, and converting that into some real world applications to help what's going on in the real world. And so I think it's fantastic that she's doing this. Just a little blurb about what she started. But, some of the products that they sell is looking at, if you go back to the previous slide, here in this white box, they're saying looking for new ways to enhance phosphate availability. So they're trying to tackle this problem of, it's hard for plants to get access to phosphorus. And so they have microbial amendments that are designed to help the plant get access to phosphorus. And then they have a bunch of other microbial amendments that are good for controlling certain plant diseases. And so they're working on this to try to create kind of a three-pronged approach to protect crops, stimulate yield. And what's the last one? Seed. Yeah, seed growth. Okay. So, there's a lot of information that's being done through academic research, but we should take it to completion. We should get it out into the real world and hopefully creating better situations for growing plants. Another idea to think about too is when it comes to phosphorus, mycorrhizal fungi are very important in that process too. It's not just the bacteria in the soil. Certainly the fungi, many different fungi have really interesting enzymes that they can use that, the plants don't have those enzymes, but the microbes do. And this is a case where it's not just the mycorrhizal fungi, but I thought this one was really cool. It's the mycorrhizal fungi. So as their hyphae are growing into areas where there's phosphate, bacteria will move along the hyphae and go to, so as the hyphae is growing, it's like a highway system for the bacteria. Has anyone heard about this? It's wild. And so as the roots get into the phosphorus abundant area of the soil, it delivers the bacteria there too. And it's the fungus and the bacteria that are there solubilizing the phosphate so that it can be taken up by the root system. And so let me see if I can, hang on. Hang on, let me see if I can get into, I don't think it'll show up in the recording, but let me just try to show it to you here. And I put the link in the notes section. This was the real-time movement. So that's the fungal hyphae, and you see the little cells moving along. Those are the bacteria. So they're using the hyphae as a tool, a highway system for them to easily move through the soil. They just there's a biofilm, there's like a slimy goo that's created on the surface of the hyphae, and it allows the bacteria to just kind of glide and move along. Well, they're both benefiting, so you could say it's a mutualistic symbiosis, and they're cooperating, if you want to use that term. Could be an example of cooperation where I think that's just fantastic, that they're able to team up, so to speak. And so they've done controlled studies where they put phosphorus in the soil in discrete patches, and then they inoculate the fungus and watch what happens. And the roots, they actually use the word forage. The roots are foraging for nutrients. They're growing and exploring, and then once they hit a patch, they'll slow down their growth and kind of stay there and feed off the phosphorus. And as they're doing that, the bacteria are moving with the hyphae through the soil.
Will the hyphae, like, let's say the plant needs phosphorus, so it'll look for it and find it, and let's say that now it has an abundance amount of phosphorus. Is it going to keep doing that process, or is it going to kind of move to the next plant?
Yeah, it's the fungus will continue to keep foraging through the soil, and it's kind of similar to what you would think. And I'm sure you've seen video footage of like a black bear with their babies moving through the forest and through the areas, and they come along a blackberry patch and they sit down and eat blackberries and then they move on. It's kind of that idea where once they've used up the nutrients in that patch, then they move on and they'll keep searching. So they actually show how roots will slow down when you provide a patch of nutrients in the soil. The roots will slow and stop growing and just sit there for a while. And then once they've used up all the nutrients, then the roots start growing again and moving out. And so roots do forage, similar to how you think about with animals foraging for their food. So
But they don't have
yeah, they'll deplete that area and then move on typically. Yeah, that's what they do. So I just thought that was fantastic footage. And so the YouTube thing is in the notes section here on this slide. So if you want to check it out again, or show your friends that aren't into plants or microbes, because it's pretty awesome. So here's another, idea again, that phosphorus is a big deal. And there's a lot of concern too among people who are in this field of study that phosphorus is becoming limiting on the planet, that our phosphorus reserves they're predicting some dire things if we don't figure this out in the next few decades forward. So it's a concern. Okay. The third, let me start. Hang on, I gotta start sharing again for the recording. So the third one in the bag of fertilizer is potassium, NPK. So potassium is pretty easy to supply if you need to. You can easily add in potash, they call it, and that is not that difficult. The downside is that if your soil is deficient in potassium is needed in very high amounts. It's used for a lot of purposes by the plant. And so the good news is it's highly soluble. So once it's into the soil system, it's easy to get it into the water solution in the soil, and the plants can take it up. And then once the plants take it up, it's really mobile through the plant body, very mobile. All right, so very or highly mobile. The only issue is that if you have sandy soil, it tends to wash through sandy soil. So, but most plants, that you would be working with aren't in sandy soil. So, symptoms. This one gives fairly definitive mottled chlorosis look to the leaves that slowly will turn necrotic over time. Also look at the very tip of the leaves and the perimeter or the margin of the leaves. Sometimes you'll see chlorosis in those spots as well. If you let the plant go too long and you don't notice that it has a potassium deficiency, and this is very famous with especially grain crops like wheat, you will notice that the plants, you'll go out to your field and the plants will have fallen over essentially. And so what happens is it ends up kind of a domino effect of low potassium can lead to stem weakening, the stem tissue weakens, and then the crops just fall over. They kind of fold over in half, and they call it lodging. And that's a problem, of course, if you are trying to harvest that wheat. Your machines don't deal with plants on the ground. And so it can be a huge issue with having to pay labor to go out there and hand harvest. It's a big issue. So this may sound like no big deal, but when you're doing acres and acres of wheat crops, you do not want a bunch of crops falling over onto the soil, just laying down. You don't want that. So again, pretty important to make sure that your potassium levels are high. Okay, so sulfur, I just wanted to mention that's really not a deficiency problem currently because we have so many pollution events that release sulfur that the soil is pretty well set for sulfur. And then the sulfur that's found in organic matter, like some of our amino acids have sulfur, microbes can degrade that quite easily. So the idea of all the pollution we're doing, lots of sulfur, microbes are releasing sulfur. So it's not common to see sulfur deficiencies. Cysteine and methionine are the two amino acids that have sulfur. And so the different coenzymes and vitamins, iso and thiocyanates contain sulfur. We'll talk more about those later in the semester. Those are interesting secondary metabolites. So when you bite into horseradish or turnips, that kind of bitter, pungent flavor that you're getting, that's the isothiocyanates. General chlorosis. So unfortunately, this is another example, like nitrogen I said was general chlorosis. Well, now sulfur is general chlorosis. So it's the same symptom. So for example, I'm not going to ask you on an exam, which of the following give general chlorosis, because there's too many. I mean, you could say all kinds of answers, and it would be challenging to set the EduCAT exam correctly. However, I might ask you on the exam, like, what does general chlorosis mean? What does that look like? Or I might show you a picture and say, what is that? And you'd have to say, oh, well, that's mottled chlorosis or something like that. All right. But asking you to memorize deficiency symptoms for every single element, I'm not going to do that. It's just, that's not the purpose. I want more that you understand the definition of, if someone says to you, your plant's chlorotic, do you know what that means? Okay. Same with immobile versus mobile. Do you know what that means inside the plant? What does that mean? Calcium's a fun one. Calcium is usually not a problem, and it's super important for the cell. More so now, we realize more and more how important calcium is. Initially, when they started looking at calcium, they were like, we're probably not going to find a whole lot. And they were very wrong. There's all kinds of reasons why calcium is needed in the plant. And a lot of it has to do with the fact that there's so-called calcium is so-called second messenger in a signal cascade system in plants. And so as plants are trying to assess their environment and then communicate that information to different parts of their body and tell the genes to turn on and off, you have to do a signaling cascade to get that to happen. You have to have this kind of domino effect of signals going through the plant body and finally getting to the target tissue, getting to the DNA. And calcium is really important in that process. And so way more than they thought. You get right at the tip of the shoot, the apical meristem, that's the shoot, you get very deformed leaves. So the new baby leaves coming out look pathetic. They look really damaged, mutated. They look really gnarly. Often they're quickly necrotic. In hydroponic systems, you'll see really poor root growth. That's visually, you'll be able to, wow, there's really little root growth. You should be thinking about calcium deficiencies. You should probably also add, in the symptom, highly stunted growth. you tend to see incredible stunted growth with calcium. Even more so, at least when I've done deficiency experiments, I notice more stunting with calcium than I do with nitrogen. And so it's quite the impact. When the plant takes it up, it is immobile. So that's why the newer baby leaves can look pathetic, because they're not going to get any calcium sent to them from the mature leaves. So the baby leaves look really bad. Okay, so calcium can be quite dramatic visually. It's fairly easy to pick up on. Magnesium is needed in high amounts. We talked a little bit about that the other day. And you can get deficiencies in either really acidic, sandy soils are an issue. It's part of chlorophyll, it's part of the ribosome, and it helps activate enzymes. I'll talk more about that, but it's absolutely necessary for rubisco. Is the most abundant protein on the face of the earth that plants have. And so hugely important for that purpose too. I told you magnesium has to be chelated to ATP. So again, magnesium is needed. This one, very obvious intraveinal chlorosis. You'll see the green veins stay green, they pop green, but the rest of the leaf go yellow, the chlorotic. And once the plant takes it up, it's very mobile. Yeah, the rubisco, it couldn't do the converting to sugar, the biochemical reaction. Yep, that would be bad if it can't make its own sugars. Here's magnesium in the chlorophyll molecule. It's chelated in the so-called head region, the porphyrin head region of the chlorophyll molecule. We'll talk more about that. Also with magnesium in the chlorophyll, I just wanted to show this comparison. Here's chlorophyll on the right, and here's hemoglobin on the left. So very similar structure. That's kind of interesting to think about. We have iron instead of magnesium. When we look at our hemoglobin, it's iron. Okay, so in the table that I asked you to look at for studying and knowing, there's also the micronutrients listed in that table. It's table 4.2, I believe, in the slide set and in the book. So again, I'm not going to ask you specific, like what symptoms does this one cause or that one cause. That's not what I'm doing on the exam. So don't waste your brain energy on that. But knowing the terminology of what are some of the symptoms to look for, what does that mean in general? I did want to say one thing about iron. Iron is a micronutrient, although some plant physiologists would argue the point it should be a macronutrient, but still it's still in the micronutrient list. It is required in the largest amounts of all the micronutrients. It's super important in the cell for many reasons. I've just listed out a couple here. Redox reactions, which are huge, going on all the time. In order to get chlorophyll fully synthesized, you need iron involved in the pathway. Intraveinal chlorosis, you'll see in the leaf. It's highly immobile. Once it gets into the body of the plant, it's going to stay put. So younger parts of the plant really start to show that intraveinal chlorosis. And then too much of a good thing. Sometimes there can be toxicity, and that can happen in humans too. It's more often than in men than women, but you can get too much iron sometimes, and that can create problems. So same with the plant, especially in acidic soils, it becomes a little too available and the plant will take up too much of it. But it's much more the case that you'll get a deficiency in iron rather than too much of it, toxicity. And so what tends to happen is that the Fe3+ form of iron, that's the ferric, F-E-R-R-I-C, ferric form of iron, it is very insoluble. And if you've ever worked with the ferric form of iron in the lab, it takes forever to get it into solution, and often we have to chelate it with something to get it into solution. And so it's just insoluble. It's really hard to work with. So you can get it into solution by chelating it to another molecule, and that will help drag it into solution, so to speak. And so in the lab, we tend to use EDTA as a chelating agent when we're mixing up a solution that needs iron. Plants, on the other hand, they can release caffeic acid. That's their chelating agent. So they will go through the trouble of synthesizing caffeic acid, secreting it out. It chelates to the iron, and then that can get brought in. And so it's a neat little strategy, rather low cost by the plant, and it allows them to get access to the iron. So this is just trying to show a visual of how it chelates the iron. Okay. And so that is a mechanism that some plants, not all, but some, can do that. So here's the structure of caffeic acid if you're curious. And so there's a couple of different strategies. Sometimes they'll release protons into the soil to get the iron more available. They'll release caffeic acid. They can, once the ferric form reaches the root, they can release an enzyme and redox reaction to get it reduced to Fe2+, and then they can take that up. And so there are some nifty little strategies that some plants have evolved because iron is so important. They've had to evolve some mechanism in order to get access to iron, even the Fe3+ iron if they have to, that's in the soil. Okay, so on Friday, I'll talk just a little bit more about iron, and then the next topic we're getting into is biological nitrogen fixation. We'll be getting into that next.